It's a question that grabs you. Can we find intact DNA from a million-year-old human? Just thinking about it gives you chills. Imagine taking a tiny piece of bone, doing some science, and pulling out the very blueprint of someone who lived a million years ago. Someone who walked the earth when things were so different, before our species even existed. It sounds like something out of a movie, right? But here's the thing. Science is amazing. We've done incredible stuff already with ancient DNA. We've found DNA from Neanderthals, from mammoths, from horses. Some of this DNA is hundreds of thousands of years old, so if we can do that, why not a million years? Why not a million-year-old human? It's a natural leap for the mind to make. And the short, honest answer is probably not. Not in any usable, intact way. Not with what we know now. The universe, in its usual way, is pretty good at breaking things down. And DNA, as precious as it is just a chemical molecule. And chemical molecules, given enough time and the right conditions, degrade. And they fall apart. So, let's talk about why. Let's look at what DNA is, how it breaks down, and what we've actually managed to find so far. It helps to understand the rules of the game first. First, what is DNA? You've heard of it. It's often called the blueprint of life. That's a good way to think about it. Every living thing has DNA inside its cells. It's like a super long instruction manual, packed with all the information needed to build and operate that organism. For us humans, it dictates everything from our eye color to how our brains develop. This instruction manual is made up of a long string of smaller chemical units called nucleotides. There are 4 types of these units, often just called letters A, T, C, and G. They link up in a specific order, and that order is the code. That code is what makes you you. That code is what makes a human, and a horse, a horse. And DNA has a famous shape, too. The double helix. 2 strands twisted around each other like a ladder. This structure is pretty strong when it's inside a living cell, actively being used and repaired. But once the cell dies, once the organism dies, the repair mechanisms stop. And that's when the trouble starts. Imagine that super long instruction manual. Now imagine it's printed on paper that slowly but surely just falls apart over time. That's what happens to DNA. It's not meant to last millions of years outside a living cell, especially not under most conditions on Earth. DNA is a delicate molecule, chemically speaking. It's constantly under attack from its environment. Think of it like an old book left out in the rain, then in the sun, then nibbled by insects. It won't stay intact for long. One of the biggest enemies of DNA is water. We need water to live, but water can slowly break down those chemical bonds that hold the DNA molecule together. It's a process called hydrolysis. Little by little, water molecules can sneak in and snip parts of the DNA chain. It doesn't happen all at once, but over hundreds of thousands of years, it adds up. These snips turn the long, unbroken DNA strands into smaller and smaller fragments. Then there's oxygen. We breathe oxygen, but it's also a highly reactive molecule. Well, oxygen can cause damage to DNA, just like it makes metal rust or an apple turn brown. It's called oxidation. This damage changes the DNA structure, making it harder to read. It's like pages in our instruction manual getting stained and smudged until you can't make out the words anymore. Oxygen can also create harmful free radicals that attack the DNA. Temperature is another huge deal. Heat speeds up chemical reactions. So, if DNA is kept warm, it falls apart much, much faster. Think about food. If you leave food out on a hot day, it spoils quickly. Put it in the fridge, it lasts longer. Put it in a freezer, it lasts a really long time. DNA is kind of similar. Cold slows down all those destructive chemical reactions that break down DNA. So, it buys the DNA time, a lot of time. Microbes are also a problem. Bacteria, fungi, tiny single-celled organisms, they love to eat organic matter. And DNA is organic matter. When an animal or person dies, these tiny organisms get to work, breaking down everything. They're like little scavengers, and they'll happily munch on DNA too. If a body isn't buried quickly or in a sterilish environment, microbes will destroy most of the DNA pretty fast. They literally consume it. Even radiation from the environment, just background radiation from the ground or space, can cause damage. It's not a huge amount at any one time, but over millions of years, those tiny zaps can add up, causing breaks and changes in the DNA. It's like tiny little nicks and tears appearing on our instruction manual pages. And then there are specific kinds of chemical damage that happen to DNA over time, even without the big, dramatic environmental factors. One common type is called deamination. This is where one of the DNA letters, specifically C for cytosine, slowly changes into a different letter, U for uracil, which then looks like T for thymine, when scientists try to read it. This happens naturally after DNA is out of a living cell, so when we try to read this ancient DNA, these changes look like real genetic mutations, but they're just post-mortem damage. It makes sequencing very tricky. We have to learn to tell the difference between a real ancient genetic change and just a chemical breakdown. So, all these things water, oxygen, heat, microbes, radiation, and even natural chemical changes octopi set around to break DNA into smaller and smaller pieces. And it's not a neat, clean break, it's messy. Imagine our instruction manual getting ripped into thousands of tiny random pieces, and then many of the letters on those pieces fading or changing. The longer the time, the smaller and more damaged the pieces become, until there's nothing left that makes sense. But wait, we have found ancient DNA. So how did that happen? It all comes down to preservation conditions. Think of these as DNA's best friends. The absolute best friend for DNA is cold. Really, really cold. Freezing temperatures slow down those destructive chemical reactions almost to a halt. This is why places like Siberia or the Arctic, with their permafrost ground that stays frozen year-round, bloom are gold mines for ancient DNA. If a creature dies and is quickly frozen and stays frozen, its DNA has a much better chance of surviving. Dryness is another big helper. Remember how water breaks down DNA, so if there's no water, or very little water, the DNA has a much better chance. Deserts, dry caves, or even very dry, ancient tombs can offer some protection. Mummification, whether natural or artificial, works partly by drying out the body. Low oxygen is also good. Since oxygen damages DNA, an environment with very little or no oxygen anaerobic conditions helps a lot. Think of bogs or deep, anoxic sediments at the bottom of a lake. These places can be surprisingly good for preserving organic material. A neutral pH level helps too. Extreme acidity or alkalinity can speed up DNA degradation. A balanced, neutral environment is generally better. And finally, the type of material the DNA is in matters. In bone, especially the dense parts like the petrous bone, a part of the skull near the ear, and teeth are much better at protecting DNA than soft tissue. Bone is like a shield. It encases the DNA and protects it from some of the immediate environmental damage. A tooth enamel, the hard outer layer of a tooth, is even better, acting like a tiny, super hard vault. So with these ideal conditions in mind, what's the oldest DNA we've ever found? The records are pretty impressive. We've found DNA from woolly mammoths, often tens of thousands of years old, sometimes pushing past 100,000. These usually come from permafrost. We've sequenced entire genomes from Neanderthals. These ancient human relatives lived in Europe and Asia. We have Neanderthal DNA dating back maybe 50,000 to 100,000 years, sometimes even older. One incredible find from a cave called Cima de los Huesos in Spain gave us Neanderthal and Denisovan-like DNA fragments. Denisovans are another ancient human group, close cousins to Neanderthals, that are around 400,000 years old. This was an amazing achievement because the cave isn't frozen permafrost, it's a cool, stable cave environment. That 400,000 year mark is a big deal, but the current world record for the oldest DNA ever sequenced comes from a horse. A more than 700,000 year old horse found in the Yukon permafrost. That's a huge number. That's pushing almost 750,000 years. This DNA was highly fragmented, like tiny puzzle pieces, but scientists managed to put enough of it together to get a picture of what that ancient horse was like. So, 700,000 years. That's pretty close to a million, right? Maybe a bit more effort, a bit more tech, and we can get to a million years. Well, it's not quite that simple. Getting to 700,000 years from, say, 100,000 years is a monumental leap. And getting from 700,000 years to a million years is another huge leap. It's not like climbing a steady ladder. It's more like the last few steps of a very, very slippery mountain, where each step becomes exponentially harder. The problem is that DNA degradation isn't a linear process. It's closer to exponential. Think of it like a decaying radioisotope or a very fast snowball melting. It loses a lot of its integrity very quickly, and then the rate slows, but the damage keeps accumulating. Every single year that passes, even in the best conditions, means more chemical bonds breaking, more bases changing, more tiny fragments. By the time you get to a million years, the amount of original usable DNA left is, to put it mildly, minuscule. It's practically nothing. Scientists have even tried to estimate a theoretical upper limit for DNA survival. Depending on the conditions, these estimates often fall somewhere between 500,000 years and 1.5 million years, with the general consensus being closer to the lower end for anything truly usable. For a human, or rather a hominin, from a million years ago, we're probably pushing right up against, or even past, that theoretical limit. Even in perfect continuous frozen conditions, the chemical bonds just keep breaking down. So, what kind of human are we even talking about from a million years ago? It's important to clarify this. Our own species, Homo sapiens, only evolved maybe 300,000 years ago, mostly in Africa. So, a million-year-old Homo sapiens is impossible. We didn't exist then. But there were other kinds of humans, or more accurately, hominins, around a million years ago. These were our direct ancestors, or close cousins. We're talking about species like Homo erectus, who had spread out of Africa into Asia and possibly Europe. We might also be talking about Homo antecessor in Europe, or early forms of Homo heidelbergensis. These were sophisticated, tool-making, fire-using hominins. They walked upright, had larger brains than earlier hominins, and were very successful. If we were to find DNA from one of them, it would be absolutely groundbreaking. It would tell us so much about their relationship to us, their migrations, their adaptations. But the challenge is immense. To find anything, we would need to hit the jackpot of jackpot preservation condition. We're talking about a million years of continuous, unbroken, perfect preservation. Imagine a hominin dying exactly a million years ago, maybe in a cave. Not just any cave, but a very cold cave. A cave that somehow stayed dry, never flooded, never warmed up, and the body, or at least a bone, had to be quickly buried in a fine anoxic sediment. Something that sealed it away from oxygen and microbes, and the bone itself had to be one of the best for DNA preservation, like a petrous bone or a dense tooth. Think about it this way, the 700,000-year-old horse DNA came from permafrost in the Yukon. That is one of the most ideal preservation environments on Earth. Most of the Earth is not like that. Most places warm up, cool down, get wet, get dry, have active microbial life. Sites where hominins lived a million years ago are mostly in warmer climates, Africa, parts of Asia, southern Europe. These places simply don't have the kind of sustained, extreme cold needed to preserve DNA for so long. The few sites in colder regions that might exist are incredibly rare, and the chances of a hominin dying there and being perfectly preserved for a million years are astronomically low. Even if we found such a fossil, say a tooth, from a million-year-old hominin, perfectly preserved in a deep, continuously frozen cave, we'd still face massive challenges. The DNA would be extremely fragmented. We're talking about pieces just a few letters long. Imagine trying to read an entire 500-page book where every page has been ripped into confetti, and then most of the confetti pieces have faded or changed their letters. It's like trying to reassemble a massive jigsaw puzzle with almost all the pieces missing, and the few pieces you have are tiny, damaged, and don't quite fit together right. Then, there's the damage. Remember deamination, those chemical changes that make C look like T. At a million years old, these changes would be rampant. It would be incredibly hard to tell what was original sequence and what was just chemical decay. It would make it very difficult to confidently reconstruct even small sections of the genome, let alone an entire one. It creates a lot of noise in the signal, and contamination. This is a huge problem with ancient DNA. Our own modern human DNA is everywhere. It's on our skin cells, our breath, our clothing. When scientists handle an ancient bone, they have to be incredibly careful not to get any of their own DNA on it. If you're trying to find minuscule amounts of million-year-old DNA, even a tiny bit of modern human contamination could completely overwhelm the signal. It's like trying to hear a whispered secret in a very loud concert hall. Also, bacteria and fungi are everywhere. Even in preserved conditions, there's often bacterial DNA present from microbes that lived on or in the bone. Filtering out all this microbial DNA from the tiny fragments of hominin DNA is another monumental task. So, while our technology for sequencing DNA has gotten incredibly good, we can now sequence very tiny amounts of DNA, and we can do it much faster and cheaper than before. It still has its limits. These technologies are constantly improving, and who knows what future breakthroughs might bring. Perhaps methods to repair heavily damaged DNA before sequencing, or even more sensitive ways to detect the specific ancient chemical signatures. But those are ideas for the future, not capabilities we have now. And what about environmental DNA, eDNA? This is DNA that can be found in soil samples or sediments, not directly from a bone or tooth. Scientists have had some amazing success with eDNA, finding traces of ancient animals and even Neanderthals and Denisovans just from digging up soil in caves where they lived. This works because living things constantly shed cell skin, hair, waste, and these cells contain DNA. This DNA then sticks to the soil particles. Could eDNA push the limit to 1 million years? For identifying the presence of a species in a given location, maybe. But eDNA also degrades. It's not protected by bone. While some DNA can cling to soil particles for a very long time in ideal conditions, getting enough intact, readable hominin DNA from soil that's 1 million years old to reconstruct a genome is likely even harder than from a bone. It's already been broken down by the elements before it even settled into the soil. We might get fragments, tiny ones, but not enough to build a picture. Why does it matter, even if it's super hard? Why do we keep pushing the limits? Because the information in DNA is unparalleled. It's the ultimate source code. If we could get DNA from 1 million-year-old hominin, it would change everything we know about human evolution. It would tell us about their genetic diversity. It would reveal migration patterns and how different groups of hominins moved across continents. We could learn about their physical traits, like skin and hair color, their resistance to diseases, their diets, and maybe even clues to their cognitive abilities. We could see how they adapted to their environments. We could understand the precise relationships between species like Homo erectus, Homo antecessor, and later hominins like us. Where did the splits happen? Who interbred with whom? The fossil record is amazing, but it can only tell us so much about the genetics of these ancient populations. DNA is the definitive link, but for now it remains a dream, a wonderful, tantalizing dream. So, to come back to the original question, can we find intact DNA from a million-year-old human? The most realistic answer, based on everything we know about DNA degradation, the conditions required for preservation, and our current technological capabilities, is no. It is exceedingly astronomically unlikely that we will ever find intact, usable DNA from a million-year-old hominin. The sheer amount of time, the relentless chemical breakdown, and the rarity of truly perfect, sustained preservation environments make it seem impossible. We might, in very rare, exceptional circumstances, push the boundary a bit past the 700,000 year mark. Maybe in another few decades, with even more advanced techniques, we might recover truly tiny, fragmented bits of DNA from a very early hominin that lived maybe 800,000 or 900,000 years ago if it died in exactly the right, freakishly perfect, deep freeze conditions. But even then, we're talking about extremely short, damaged pieces, not an intact genome that would give us the full picture. The current scientific consensus suggests that for any meaningful DNA information, the million-year barrier is likely impassable. DNA just doesn't last that long. It's a chemical reality. But this isn't to say that the work being done on ancient DNA isn't incredible. We have learned so much from DNA that is hundreds of thousands of years old. We've rewritten parts of human history, understood our connections to Neanderthals and Denisovans, and even brought back details about ancient ecosystems. The quest for older and older DNA will continue. Scientists will keep trying because that's what science does. It pushes boundaries, it tests limits, and who knows what truly unforeseen discovery might be out there. A hominin fossil preserved in a totally unique, never-before-seen environment. A new chemical stabilization mechanism we don't understand yet, but for now, the dream of a million-year-old human genome remains just that, a dream. A fascinating one, but a dream nonetheless. The universe has its ways of keeping its oldest secrets.